Hello, my name is Abayisenga Pacific from Ines Ruhengeri. I'm doing biomedical laboratory science in year 3 and also I'm in group 1. My reg number is 23/20.240 and I would like to present to you the 5 questions which start from question number 33 up to 37. So without further delaying, let's kick off on the first question. So the first question is, what is the principle of precipitation reaction in immunology? So phase 3 here we are going to see what is it, precipitation reactions in immunology and its principle. So this precipitation reaction is going to occur when there is a soluble antigens which is going to bind to the antibody, the specific antibody, and forming an insoluble complex which is an antigen antibody complex. The principle of this precipitation reaction in immunology, when the antigens and antibodies they come together, they form a lattice-like structure which precipitate out of the solution. We are going to see on the picture how this lattice-like structure is going to be formed, and the point where the antigen antibodies they are going to meet is called the zone of equivalence. So as you see, we have an antibody at the bottom and an insoluble antigens at the top, and this precipitation is where the antigen and antibody they are going to form a complex and there is where we are going to see the zone of equivalence. As you see, the zone of equivalence where we are going to see the visible precipitate. So let's move on the next question, which is the radial immunodiffusion test performed. How radial immunodiffusion test performed? Radial immunodiffusion quantify the specific protein is the sample using antigen antibody precipitation in a gel. Here we are going to see how to perform this radial immunodiffusion by steps. The radial immunodiffusion test is performed by the following steps, as you are going to see. The first step is to prepare the gel, where this gel is going to be prepared. Here the gel we are going to use is agarose gel, and it's going to be mixed with antiserum. After the solution is going to be poured into the container, either petri dish, and is going to be allowed to solidify. So the next step is cut wells, is where we are going to put the whole or wells into the gel. You know, the solidified gel we are going to put into the holes on the gel, followed by the third steps, which is to add the sample, where the wells is going to be filled with the sample containing antigens. The next step is going to be cover, where we are going to cover our petri dish or slide to prevent the evaporation. The next step is to incubate, where we are going to incubate our petri dish or slide at 37 degrees Celsius. Here we may use a slide or a petri dish. As you know, in the microbiology we used to use a petri dish in culturing. Here we may use a petri dish in this manner. The next step is measuring, where we are going to measure the diameter of the ring which is going to be formed. It is called precipitating ring, and this ring is going to be formed, and we are going to measure its diameter, and its diameter is going to correspond to the concentration of antigen in the sample. So the last step is to compare, where we are going to compare the diameter of the sample ring to the diameter of the ring formed by the known concentration of the antigens. And all of those steps are those steps for these procedures. And let's move on the next question, which is, what is the principle of ELISA technique? As we know, ELISA stands for enzyme-linked immunosorbent assay, which detects and quantifies antigens or antibodies in the sample using enzyme-conjugated antibodies. and the calorimetric reaction. This calorimetric reaction is that reaction which is going to be between an enzyme and a substrate and then produce a color, a visible color, which is homogenic, which is a color which is going to be formed due to the fusion of enzyme and its substrate. So let's move on the principle where we are going to see this principle of ELISA, which is based on specific binding of antibodies or antigens. Their target antigens means the antibody is going to bind to their specific antigens, to their target antigens, so that which is going to be followed by the enzyme linked antibodies, which is going to be conjugated with antibodies, which is going to be conjugated with an enzyme, and we are going to add the substrate after and quantify the antigens binding by quantifying their colors. So let's move on the next question, which is sandwich ELISA. How sandwich ELISA different from indirect ELISA? Here we are going to consider the features, those features: coating, primary binding, primary use, and detection for differentiating sandwich ELISA and indirect ELISA. Firstly, for coating, we are going to see the plate all aware, which is going to be coated with a capture antibodies on sandwich, wherefore indirect, the plate all aware is going to be coated with an antigen. So for primary binding on sandwich ELISA, we are going to see the antigen which is going to bind to the capture antibodies, because the plate was coated primary with an antibody. The antigen is going to be the one which is going to bind first. On indirect ELISA, as the well is coated, or plate is coated with an antigen, the primary antibody is the one which is going to follow. For primary use of sandwich ELISA is to detect and quantify the antigens. As the well or plates were coated with an antibody, those antibodies, they are going to make reactions with an antigens, and we are going to detect those antigens and quantify them. Indirect ELISA, we are going to detect and quantify antibodies, as we have seen that the well or plate is going to be coated with an antigen, and we are going to quantify the antibodies which are going to bind to those antigens. For detection, this sandwich ELISA use a pair of antibodies, where those antibodies, they are specific to different epitopes of the target antigens. And for indirect ELISA, they are going to use the primary antibodies, as we have seen for primary binding, which is going to be followed by secondary antibodies, which is going to be specific to that primary antibody. It is going to be specific to this primary antibody, and that secondary antibody is going to be conjugated with an enzyme which is going to react with a substrate and give a visible color. As you see here, there is a diagram showing direct ELISA and a sandwich ELISA. For indirect ELISA, we have seen that the well or a plate is going to be coated with an antigens, which is going to be followed by primary binding of this primary antibody, followed by these secondary antibodies, which is conjugated with enzymes, which is going to react with those substrates. For sandwich ELISA, the well or a plate is going to be coated with an antibody, where the primary binding is going to be an antigens, and antigens is going to be followed by an antibody. For this sandwich ELISA, this antibody may be conjugated with an enzyme, or may be followed by another antibody which is conjugated with an enzyme. And when this antigen is followed by an antibody conjugated with an enzyme, is going to be direct sandwich ELISA, and when is followed by another antibodies which is conjugated with an enzyme, is going to be an indirect sandwich ELISA. Let's move on the next question, which is what are the applications of ELISA in clinical practices? So this ELISA is widely used in diagnostics and research and therapeutic monitoring. We are going to see the applications here, where the first application is for infectious disease diagnosis, where this ELISA is going to detect antigens or antibodies for a disease like HIV, hepatitis, and COVID-19. The next application is hormone measurements. This ELISA is going to be used for monitoring hormone levels such as TSH and HCG, where TSH is going it stands for thyroid stimulating hormone, and HCG stands for human choreogonadotropic hormone. And the next application is for autoimmune disease detection, where this ELISA is going to detect those autoantibodies, where, as we know, autoimmune disease, they are those diseases caused by those autoantibodies which mistakenly attack their own immune cells, where this ELISA is going to identify those autoantibodies and, for example, those rheumatoid factors. The next application is going to be allergy testing, where this ELISA is going to be used in measuring allergen-specific IgE antibodies, and the next application is going to be cancer markers, where this ELISA is going to quantify the tumor markers, such as those markers PSA, CA125. Those are tumor markers, and this ELISA is going to detect and quantify those markers. The next application is therapeutic drug monitoring, where this ELISA is going to track drug levels such as therapeutic antibodies, those drugs which contain those therapeutic antibodies. And the last application is in research, where this ELISA is going to be used in the research. In measuring, such as measuring cytokines, growth factors, and other biomarkers. So thank you for your kind attention. I hope you have enjoyed the presentations. You have catch something. Thank you very much. May God bless you.